In the previous video, we discussed about hypersensitivity in general. If you want to watch that video first, the link is in the description. Now here we are going to discuss about hypersensitivity type 1. The hypersensitivity type 1, aka immediate or anaphylactic hypersensitivity. This is an exaggerated immune response that occurs within minutes of exposure to an allergen, but only in individuals who have been previously sensitized. That means the person's immune system has already encountered that allergen before and produced antibodies against it. This type of hypersensitivity is mediated by IgE antibodies. The main effector cells involved are mast cells and basophils, and the onset of reaction is very fast, usually within seconds to minutes after exposure. Furthermore, major chemical mediators are like histamine, leukotrienes, and prostaglandins. These chemical mediators cause the typical allergic reactions such as vasodilation, increased vascular permeability, and smooth muscle contraction. Some common examples include most allergic reactions, asthma, food allergies, pollen allergy, and even severe reactions like bee sting, anaphylaxis, and many more. Now let's understand how type 1 hypersensitivity actually happens. The mechanism can be divided into two main phases: the sensitization phase and the effector phase. Here in this diagram we have an allergen, and this is the first time the body is encountering such an allergen. So the first exposure to an allergen is known as sensitization phase. The immune system doesn't cause any visible allergic reactions. Instead, the allergen is recognized by our immune system and IgE antibodies are produced. These IgE molecules then bind to the mast cells and basophils, coating their surface and preparing the body for future reactions. Then comes the subsequent exposure. This is the effector phase. Now when the same allergen enters the body again, it binds directly to those IgE antibodies that are already sitting on the mast cells and basophils. This triggers these cells to degranulate, releasing powerful chemical mediators like histamine, leukotrienes, and prostaglandins, leading to the classic allergic symptoms we see. So remember, the first exposure only sensitizes the body, while the second exposure triggers the allergic reaction. Now let's see the whole mechanism in detail with animations. We see in the diagram we have the epithelial barrier. Inside it we have the dendritic cells. First of all, allergen comes in contact with the epithelial barrier as shown in the diagram. It either sneaks through the barrier or releases allergen components as shown in the diagram. These allergen components are perceived by dendritic cell via TLR and CLR. The dendritic cell internalizes the allergen by endocytosis, where the allergen is processed and finally presented on MSC class II molecule on dendritic cell surface. After that, dendritic cell migrate to the lymph node as shown in the animation. Then this mature dendritic cell presents the allergen to CD4 plus T cell, that's Th0 cell. Interaction occurs as shown in the diagram. TCR of T cell interacts with MSC2 and CD8T with CD28. Due to this interaction, cytokines are released, particularly the IL-4. But we see some other cytokines also like IL-6 and IL-10. But IL-4 is important here because it polarizes the T cells and differentiates Th0 cell into Th2 cell. After this differentiation, we see the Th2 cell secretes different types of cytokines. Like we see high levels of IL-4, IL-5, IL-9, IL-10, IL-13. And among these cytokines produced, the IL-4 and IL-13 mediates the B cell switching, which is one of the important steps here. So we see here IL-4 and IL-13 goes to the B cell and mediates the B cell switching. So after the B cell switching, we see the B cells are made to secrete IgE antibodies as shown in the animation. So now we have high levels of IgE antibodies present within the circulation. Now these IgE antibodies searches for the mast cell or basophils. So here in this animation we can see IgE antibodies binds with the receptors of mast cells. Once this happens, the mast cells are said to be armored or sensitized. So we see in this diagram the mast cells are loaded with IgE antibodies. So these are the armored mast cells. Now we move on to the second phase, that's effector phase. That's when there is exposure to the same allergen. Now when the same allergen enters the body again, the allergen cross-links two or more IgE molecules bound on the mast cells as shown in the animation. So this cross-linking triggers intracellular signaling within the mast cells, leading to degranulation. So upon degranulation, a cascade of chemical mediators like histamine, leukotrienes, prostaglandins is released into the surrounding tissue. These mediators are responsible for the classic allergic symptoms like vasodilation, increased vascular permeability, smooth muscle contraction, and intense itching. The immediate phase, which occurs within the minutes, leading to swelling, redness, and in severe cases like anaphylaxis. A few hours later, the late phase reaction begins, characterized by infiltration of eosinophils, basophils, and Th2 cells, which sustain inflammation and cause tissue damage if the response continues. So this is what type 1 hypersensitivity is. I hope you like the video. If you like it, give it a thumbs up. Consider supporting my work on Patreon or YouTube and make sure to subscribe this channel. Thanks.